Welcome to BioBits and Bytes. Have you ever stopped to think about the sheer miracle of how we go from a single microscopic cell to the complex beings we are,
It's pretty mind-blowing when you consider it.
right? Or the incredible journey life has taken over billions of years to become so diverse?
Absolutely. Two sides of the same incredible story.
Today, we're diving deep into that fascinating interplay between developmental biology and evolution.
Sounds great.
We've got a rich stack of material here, a comprehensive textbook and a cutting-edge scientific journal, and our mission is, well, to unpack the key steps in how a single cell builds an entire organism.
And the evolutionary forces that have sculpted those processes.
Exactly. And to help us navigate this intricate landscape, we have a brilliant scientist joining us. You're going to help shed light on some of the key scientific terms, right?
I'll certainly do my best to define those terms clearly. It's wonderful to be here, ready to explore these captivating aspects of life's journey with you.
Fantastic. Let's jump right in, shall we? Our sources begin at the very beginning. Fertilization. This marks the start of an extraordinary transformation.
It really does. From that single cell, everything else follows.
Can you walk us through this initial stage? What's happening at the cellular level?
Absolutely. Fertilization is that pivotal moment when a sperm and an egg fuse. This creates the very first cell of a new organism, the zygote.
The zygote. Okay.
And this single cell is described as diploid. Could you explain diploid for us?
Good point, yes. Diploid means it contains two complete sets of chromosomes.
Right, one set inherited from each parent. Now, the sperm and egg themselves, before they fuse, are haploid.
And haploid means?
Haploid means they carry only one set of chromosomes each. Think of it like two halves coming together to make a whole.
Okay, got it.
And the textbook emphasizes that the journey from forming these haploid gametes to the diploid zygote is really carefully orchestrated across generations.
To keep the chromosome number consistent.
Exactly. It ensures that number remains stable.
So from that single diploid cell, this whole organism unfolds. Our sources detail two key phases after that, embryogenesis and the fetal period. What are the critical distinctions there?
That's a great question. Embryogenesis. Maybe you could define that one first.
Sure. So embryogenesis, also sometimes called organogenesis, is essentially the initial blueprint phase.
Right. In humans, this covers roughly the first 8 weeks of development. It's a really intense period where the groundwork for all the major organs and body structures is laid down.
We call those organ primordia.
Yes, the organ primordia. It's a flurry of cellular activity, cells dividing, moving, starting to specialize.
And organogenesis specifically refers to that organ formation part.
That's right. Organogenesis is the process of forming those organ primordia during embryogenesis.
Okay. And after embryogenesis.
Following that foundational phase is the fetal period. This goes from the end of the 8th week right up until birth.
Well, what's happening then? Left blueprint? More.
Yeah.
Eating.
Exactly. In the fetal period, the organs continue to refine their structure and function, becoming more specialized. Plus, the fetus undergoes significant growth in size and mass, getting ready for the outside world.
It's really astonishing how our understanding of this has grown. The textbook mentions pioneers like Jacques Loeb, J. Holtfrieder, G.G. Pincus. What were some of their big contributions?
Oh, these were true visionaries. Jacques Loeb, for instance, around the turn of the 20th century, did something remarkable. He induced artificial parthenogenesis in sea urchin and frog eggs.
Parthenogenesis. What is that exactly?
Parthenogenesis is the development of an egg without being fertilized by sperm.
Wow. So he showed the egg could start developing on its own.
Sort of. Loeb's work really cracked open the idea that the egg itself holds a lot of developmental potential, maybe more than people thought, independent of the sperm's genetic contribution.
Interesting. And Holtfrieder.
Later, in the 1930s, Holtfrieder introduced the concept of the organizer. Could you explain the organizer?
An organizer is a specific region in the early embryo that can, well, direct the development of the tissues around it.
Precisely. And tied to that is the idea of inducers.
Which are?
Inducers are the signals or substances released by these organizer regions. They essentially tell other cells what to become, like a conductor leading an orchestra of cells, you know.
That's a great analogy. And Pincus.
G.G. Pincus, also in the 1930s, achieved a big milestone by artificially inducing parthenogenesis in a mammal, a rabbit.
So taking Loeb's work into mammals.
Exactly. These experiments, taken together, really demonstrated the active and intricate roles of different embryonic parts in guiding development. It wasn't just a passive unfolding.
Building on that, the textbook talks about developmental model organisms.
Right.
Why rely so much on, like, fruit flies and zebrafish.
Ah, model organisms. That's a crucial concept. Maybe you can define what makes something a model organism.
Sure. Model organism is basically a species researchers choose because it has characteristics that make it really suitable for studying specific biological questions.
Right. Things like a short generation time, so you can see changes across generations quickly, or a genome that's well understood and relatively easy to manipulate.
And the textbook lists a whole range, right?
Yeah.
Plants, worms, flies, mice, even axolotls.
Yes. Arabidopsis, Drosophila, C. elegans, sea urchins, ascidians, xenopus frogs, zebrafish, donio, chickens, gallus, mice, axolotls, planarian worms. It's a diverse list. Some, like mice, share significant similarities with humans, making them vital for disease research.
But the key insight is conservation, isn't it?
Absolutely. Despite the incredible diversity out there, many fundamental developmental mechanisms are remarkably conserved, meaning they're similar across very different animals.
So studying a fly's development tells us something about our own.
Precisely. It helps us decipher universal principles without the ethical or practical issues of studying human embryos directly.
Okay, let's zoom in on the sperm cell itself. The textbook has some detailed illustrations. Can you walk us through the key parts?
Certainly. The sperm is a marvel of biological engineering, designed for one mission: reach and fertilize the egg.
It's got distinct parts.
Yes, a head, a neck, a middle piece, and a long tail. The head is key. It contains the nucleus, and the nucleus holds the chromosomes. Could you remind us what chromosomes are?
Chromosomes are the structures within the nucleus that carry the genetic information, the DNA, organized into genes.
Exactly, the genetic blueprint from the father. Covering the front of the head is a specialized cap called the acrosome.
The acrosome. What's its job?
Think of the acrosome like a tiny drill bit or maybe a pack of enzymes. These enzymes are essential for the sperm to break through the egg's outer protective layers.
Okay. Then the neck and middle piece.
The neck connects the head to the middle piece, and the middle piece is absolutely packed with mitochondria.
Mitochondria being.
Mitochondria are the powerhouses of the cell. They generate the energy, the ATP, needed for the tail to beat so vigorously.
And the tail provides the movement.
Right. The tail, or flagellum, drives the sperm's motility, propelling it towards the egg.
So these gametes, sperm and ova, carry our hereditary info. How are they actually made?
That process is called gametogenesis. It's a specialized type of cell division. In males, it's spermatogenesis in the testes, making sperm. In females, oogenesis in the ovaries, making ova or eggs.
And the key is getting the chromosome number right.
Exactly. You start with diploid cells, but through a process called meiosis, you end up with haploid gametes. Having the chromosomes is essential so that when sperm and egg fuse, the diploid number is restored in the zygote. Keeps things stable across generations.
Okay, the sperm's made it. What happens at that critical moment of fertilization? Sounds like it's more complex than just bumping into the egg.
You're absolutely right. It's a sophisticated sequence. Our early understanding, the textbook notes, came a lot from invertebrates like sea urchins.
What happens there?
The sperm binds directly to the egg surface, triggers that acrosome reaction releasing enzymes, and forms an acrosomal process, a little projection to help penetrate.
But it's different in mammals.
It is. We have an extra barrier. The zona pellucida. Can you define the zona pellucida?
The zona pellucida is a thick, sort of sugary protein layer surrounding the mammalian egg. It's a protective coat.
Exactly. And the sperm has to get through that, which leads to two key processes in mammals mentioned in the text: capacitation and the acrosome reaction.
Okay, let's break those down. Capacitation first.
So capacitation is a series of biochemical and physiological changes the sperm undergoes after entering the female reproductive tract. It's like an activation process.
What triggers it?
It's complex, influenced by the environment in the tract, involving things like changes in intracellular calcium.
Right.
Basically, it primes the sperm, makes it hypermotile, and gets it ready for the next step.
Which is the acrosome reaction.
Right. The acrosome reaction is the actual release of those enzymes from the acrosome.
When does that happen?
It happens after the capacitated sperm binds to the zona pellucida. Those enzymes are crucial for digesting a path through the zona pellucida, so the sperm can finally reach and fuse with the egg's membrane, the ulema.
So no capacitation, no acrosome reaction, no fertilization in mammals.
Pretty much. Both are absolutely essential steps.
Okay, fertilization achieved. The zygote then starts dividing rapidly. The sources mention cleavage and gastrulation. What are these early processes?
Right after fertilization, the zygote undergoes cleavage. Can you define cleavage?
Cleavage refers to those initial rapid cell divisions of the zygote.
Yes. And what's interesting is that the overall size of the embryo doesn't increase much during cleavage. The big zygote just gets partitioned into many smaller cells called blastomeres.
Eventually forming a structure called a blastula.
Exactly. A blastula, often a hollow ball of cells. Following cleavage comes a really dramatic phase called gastrulation.
And gastrulations is.
Gastrulation typically happens in the late blastula. It involves massive rearrangements, migrations of cells. It's a huge transformation.
And the result is the germ layers.
Yes, this is where the three primary germ layers, ectoderm, mesoderm, and endoderm, are established. These are the foundational tissues that will eventually give rise to all the different tissues and organs.
The textbook also mentions chemodifferentiation. What's that?
Chemodifferentiation refers to physiological changes happening in localized areas. Cells start acquiring distinct characteristics, which limits their potency to become other cell types. They're starting to commit to specific fates.
These cell movements during gastrulation physically shape the embryo. That's morphogenesis, right?
Hmm.
The book highlights epibly. Can you explain epibly?
Certainly, morphogenesis is the whole process of shaping the organism. Epibly is one type of morphogenetic movement. Can you give us the definition?
Epibly is when a sheet of cells expands and spreads out to cover underlying tissues or structures.
That's a good way to put it. Imagine stretching a sheet over a ball. The textbook often uses the example of the ectoderm spreading over the yolk sac in fish or amphibian embryos. The cells migrate and divide, thinning out as they cover a larger area.
Another crucial part of morphogenesis is how cells sort themselves out. The book discusses differential cell affinity. What does that mean?
Differential cell affinity. It describes the remarkable ability of cells to recognize and preferentially stick to other cells of the same type.
Like finding their own kind.
Exactly. It's like cells having preferences for who they hang out with. This selective sticking is vital for organizing cells into tissues and organs.
How do they do that?
Through cell adhesion molecules on their surfaces. A key family mention is the cadherins. Maybe you could define cadherins.
Cadherins are a type of cell adhesion molecule that requires calcium to function. They mediate cell-to-cell adhesion by binding to other cadherins on adjacent cells.
Perfect. And inside the cell, these cadherins are linked to proteins called catenins.
And catenins are.
Catenins are intracellular proteins that link the cadherins to the cell's internal skeleton, specifically the actin cytoskeleton. This whole cadherin-catenin complex forms structures called adherens junctions.
Which physically connects the cells.
Yes, and provides structural integrity. Different cell types express different types or amounts of cadherins, allowing them to sort themselves out and form distinct tissue boundaries during development. It's quite elegant.
Let's switch gears to the architects, genes. The book links Mendel's form-building elements to our modern view. How did that understanding evolve?
It's a fascinating story. Mendel identified heritable units but didn't know what or where they were. Later, in the late 1800s, a big debate arose. Was it the nucleus or the cytoplasm controlling inheritance and development?
Who are the key players?
Scientists like Edmund Beecher Wilson and Theodore Boveri championed the nucleus, specifically the chromatin inside, which we now know is mostly DNA. They pointed to observations of cell division, fertilization, regeneration.
All pointing to the nucleus.
Yes, suggesting the nucleus carried the essential instructions. This led to the huge realization that inheritance wasn't mystical, but involved the physical transmission of a chemical compound DNA from parent to offspring.
The book then discusses nuclear transplantation. How did this technique, from early ideas to Dolly the sheep, illuminate the nucleus's role?
Nuclear transplantation, or somatic cell nuclear transfer, cloning. It's a powerful technique. You take the nucleus from a donor cell, often a specialized somatic cell, and put it into an egg cell that's had its own nucleus removed.
And the idea wasn't new.
Oh, Yves Delage proposed it way back. Hans Spemann suggested key experiments, but Thomas King and Robert Briggs got the first real success in frogs.
And then came Dolly.
Right. In 1996, Ian Wilmot and colleagues cloned Dolly the sheep from an adult udder cell nucleus. This was landmark.
Why so significant?
It definitively showed that the nucleus of a differentiated adult cell still contained all the genetic information needed to direct the development of a whole new organism. It powerfully confirmed the central role of the nucleus in DNA.
But sometimes development goes wrong. The book introduces teratology. What is teratology and what are teratogens?
Teratology is the scientific study of birth defects, their origins, causes, mechanisms. A teratogen is any agent, could be a chemical, a virus, radiation, even a metabolic condition that can interfere with normal development and cause abnormalities.
The book gives examples.
Yes, two prominent ones. First, fetal alcohol spectrum disorder, or FASD. Alcohol is a potent teratogen.
How does it cause problems?
It can disrupt many processes: cell migration, division, adhesion, survival. Research suggests it can downregulate a key gene called sonic hedgehog, which is vital for facial and brain development.
And the other example?
Retinoic acid, a vitamin A derivative. It's crucial for normal development in the right amounts: axis formation, jaw, heart.
But too much is bad.
Yes, excessive amounts during pregnancy act as a teratogen, causing severe defects, possibly by messing with neural crest cell migration. And the book also notes maternal metabolic conditions. Autoimmune issues, diabetes, poor nutrition can also act as teratogens.
Okay, on a more positive note, the sources mention assisted reproductive technology, or RT. What does RT cover?
Assisted reproductive technology, RT, is a broad term for medical procedures helping with infertility. The best known is probably IVF, in vitro fertilization. Can you define IVF?
IVF is where eggs are fertilized by sperm outside the body in glass, and then the resulting embryo is transferred to the uterus.
Exactly. ART also includes using donor eggs, donor sperm, frozen embryos, and surrogacy is often included,
Surrogacy being.
where another woman carries a pregnancy for the intended parents. The textbook also specifically highlights gamete cryopreservation, especially sperm banking.
Which is freezing sperm. Could you define cryopreservation and sperm banking?
Certainly. Cryopreservation is the process of preserving cells, tissues, or organs by cooling them to very low temperatures, effectively stopping biological activity. Sperm banking is the specific application of cryopreservation to store sperm for future use, preserving fertility.
Freezing cells sounds tricky. How do they avoid damage?
That's where cryoprotectants come in. These are substances added before freezing. What's the role of a cryoprotectant?
A cryoprotectant helps protect cells from damage caused by ice crystal formation during freezing.
Precisely. They affect how water behaves, preventing lethal ice inside the cells. The textbook mentions two main methods: slow freezing and vitrification.
How do they differ? Slow freezing is.
Slow freezing uses lower concentrations of cryoprotectants and a gradual temperature drop, allowing cells to dehydrate somewhat before freezing.
And vitrification.
Vitrification uses very high concentrations of cryoprotectants and ultra-rapid cooling. The goal is to solidify the solution into a glass-like state without forming any ice crystals at all.
Fascinating technology. The book also briefly mentions gene knock-in. What's that used for?
Gene knock-in is a genetic engineering technique. It lets scientists insert a specific DNA sequence, maybe a mutated gene causing a disease, or a reporter gene, to track activity into a precise spot in an organism's genome, usually a mouse.
To create disease models.
Exactly. Great for studying disease progression in vivo in the living animal and understanding protein function in context.
Are there limitations?
Oh, yes. It can be complex, especially with multiple insertions. And finding reliable genomic locations is still a challenge. Plus, the big one, mouse physiology isn't identical to human physiology.
So a disease model might not perfectly replicate the human condition.
Right. The book uses the example of cystic fibrosis. The mutation doesn't cause the same severe lung issues in mice as in humans. It's a limitation to keep in mind.
Okay, let's shift now to the big picture, evolution. The textbook contrasts Lamarckism and Darwinism. Can you outline Lamarck's theory?
Sure. Lamarckism, from Jean-Baptiste Lamarck in the early 19th century, was an early evolutionary theory. Can you summarize its main ideas?
Lamarck proposed the law of use and disuse. Parts used more get stronger, parts unused weaken. And crucially, the inheritance of acquired characteristics, changes acquired during life, are passed to offspring.
That's the core of it. He thought organisms strive for complexity, environment influences habits, and these acquired traits are heritable.
But it didn't hold up, did it? What were the criticisms?
The biggest issue was the inheritance of acquired characteristics. There is just no good evidence that changes to body cells, somatic cells, could be passed down through the germline, through sperm and eggs.
Like a bodybuilder's muscles aren't inherited.
Exactly. As genetics developed, it became clear heredity worked through genes in germ cells, not acquired somatic changes. That fundamental flaw led to Lamarckism being largely rejected.
Which brings us to Darwin. His theory of evolution by natural selection. What are the core principles of Darwinism?
Darwinism, summed up as descent with modification, relies on natural selection. Could you define natural selection?
Natural selection is the process where organisms with traits better suited to their environment tend to survive and reproduce more, passing those advantageous traits to their offspring.
Perfect. Darwin observed variation within populations, that some variation is heritable, and that there's a struggle for existence because more offspring are produced than can survive.
So nature selects the winners.
In a sense, individuals with advantageous variations are more likely to survive and reproduce. Over generations, this leads to populations adapting to their environment and eventually, potentially, to speciation.
And speciation is.
Speciation is the evolutionary process by which new biological species arise.
The textbook mentions different patterns of selection, like progressive or directional selection. What's that?
Progressive or directional selection happens when the environment changes, favoring individuals at one extreme of a trait range. This shifts the population's average trait value over time.
The peppered moth example.
That's the classic case. Industrial pollution darkened trees, favoring dark moths over light ones, dramatically changing the population's color frequency.
And how does selection lead to new species? The book talks about different modes of speciation, like allopatric and sympatric.
Right. Allopatric speciation is probably the most common. Could you define allopatric speciation?
Allopatric speciation occurs when a population is geographically divided, preventing gene flow. The isolated groups then diverge genetically over time due to different pressures and mutations.
Exactly. If they diverge enough to become reproductively isolated, meaning they can't interbreed successfully anymore, they're considered new species.
And sympatric speciation.
Sympatric speciation is the evolution of new species within the same geographic area. Define that one for us.
Sympatric speciation is less common and happens without geographic separation, often through mechanisms like rapid reproductive isolation, maybe due to changes in chromosome number like polyploidy, especially in plants.
Yes, polyploidy is a key mechanism there. The textbook also briefly mentions parapatric speciation, adjacent populations with limited gene flow, and parapatric speciation, a small peripheral group buds off.
So multiple ways new species can arise. The book then moves to molecular evidence for evolution, discussing molecular phylogenetics. How does studying molecules help?
Molecular phylogenetics uses molecular data, DNA, protein sequences to figure out evolutionary relationships. Can you define molecular phylogenetics?
It's the branch of phylogeny that analyzes hereditary molecular differences, mainly in DNA sequences, to gain information on an organism's evolutionary relationships.
Right. Even before widespread sequencing, techniques like DNA hybridization gave clues, but sequencing revolutionized it.
How's it work now?
Scientists compare gene or genome sequences across species. Using statistics and computational methods, they build phylogenetic trees, also called dendrograms.
A phylogenetic tree or dendrogram being.
It's a branching diagram showing the inferred evolutionary relationships among various species based upon similarities and differences in their physical or genetic characteristics. Groups sharing a more recent common ancestor form a clade.
And a clade is?
A clade is a group of organisms believed to have evolved from a common ancestor. Molecular phylogenetics uses tools for sequence assembly, alignment, modeling sequence evolution, and then reconstruction methods like maximum likelihood or Bayesian inference to build these trees. It gives us a really detailed map of life's history.
Finally, the journal article dives deep into sonic hedgehog signaling. Why is she so important in development?
The sonic hedgehog pathway is a crucial signaling system. It acts as a morphogen, especially in the developing neural tube.
A morphogen meaning it provides positional information.
Exactly. Cells respond differently depending on the concentration of shush they receive. Shush is secreted from a source, creating a gradient. This gradient tells cells along the dorsal ventral axis of the neural tube what type of neuron to become.
So changes in spread are a big deal.
Huge. The article highlights how disrupting the normal spread may be interfering with negative regulators like TIT1 and HIP1 that usually limit its range, can mess up the pattern, expanding ventral fates.
And changing how shush diffuses also matters.
Yes. The article mentions experiments with modified shush that can't be tethered by cholesterol. This changes the gradient shape and compresses the pattern of cell fates. It also discusses other proteins like CETO and GAS1 that fine-tune the cell's response.
And forms clusters. Punctae.
Interestingly, yes. Recent work shows protein-forming punctae associated with primary cilia and microtubules in the neuroepithelium. This suggests maybe more complex transport mechanisms are involved.
And this connects back to birth defects.
Absolutely. Is critical for establishing midline structures, like in the face. As we mentioned with teratogens, downregulating maybe due to alcohol exposure, is linked to conditions like FASD and associated facial abnormalities. It's a really key developmental pathway.
Wow, we've really covered a lot of ground today. From that single cell through the intricacies of development to the grand sweep of evolution and the molecular details, like, it's incredible stuff.
It truly is uncovering the mechanisms that build life and the forces that shape its diversity. It's just fascinating.
The fields of developmental biology and evolution are clearly constantly evolving themselves, revealing new layers.
Absolutely. And what always strikes me is the interconnection. Development is evolution's product, and studying development informs how we understand evolution. It's a continuous feedback loop.
A very elegant loop. This has been a truly illuminating deep dive. Thank you so much for sharing your expertise and clarifying all those terms for us.
My pleasure. It was great discussing it.
And thank you, our listeners, for joining us on Bio Bits and Bytes.